Biggest thing that we want to talk about today is Varroa mites. So how many people here have honeybees? How many of you have Varroa mites?
Oh, not me.
Okay, if you didn't raise your hand, you're a liar. But the thing is though, you have Varroa mites not because you're a bad beekeeper, you have Varroa mites because you have bees. I don't care who you are, whether you're the best beekeeper, quote unquote, self-described. Everyone calls themselves the best beekeeper, but no one's really that good. But there's always something new to learn. But I don't care who you are, whether you run 100,000 colonies across the country or you have 3 in your backyard, you're gonna have Varroa mites. There's no questions about it. It's kind of like you have a dog or a cat, right? And they get fleas or ticks, right? What do you do if they get fleas or ticks? You give them a bath, you get a treatment, you go see a vet, you get something in them to get rid of those fleas and ticks and whatever things that they're affected with. Honeybees are really no different. They're pets, they're livestock, and they need veterinary interventions. And one of the biggest things that we could do are with these stupid Varroa mites. They're the worst things out there. We're showing that most of the reasons why people's colonies die across the country, whether you're a backyard beekeeper or commercial beekeeper, it's this parasitic mite. I can't understate how important this is. I'm not trying to scare you or say Chicken Little kind of thing, but this is just what the data say. We have a lot of research showing it. So today's what we're going to talk about is how to find Varroa mites in your colonies, what you can do to treat for Varroa mites with miticides and things like that, but then also to other more integrated measurements such as using Varroa resistant honeybee stocks are just all these things you could do to kind of keep Varroa mites low. And so today I'm gonna be talking a lot, but I want this to be more of a discussion. So raise your hand, interject, ask questions, and we'll kind of have a little discussion here as opposed to me just lecturing. So Varroa mites, what are they? What do they look like? So you can see here on the adult honeybee, this big red dot, relatively big red dot, is a parasitic Varroa mite. It has one of the largest body size to host ratios of any parasite in the animal kingdom. So this is actually very large. If it was on you, it would be about the size of a basketball. And you can see specifically where it's located here on the abdomen of the adult bee. Here's a nice close-up showing that the female Varroa mites are this football orange brown kind of thing that you can see here. And then even too, you'll see them on the pupae. So you can see this purple eye pupae here covered with a few Varroa mites. So that's what they look like, these football, red, orange, brown, kind of football looking shaped things that are parasites on honeybees. And so let's talk a little bit about their life cycle here. So as the queen's laying eggs, and those eggs hatch and become larvae, and right before that larvae turns into a pupa, that's when that Varroa mite will infest the cell. She gets in there, and then that cell is capped. Once that cell is capped, after a certain period of time, that female mite that entered the cell will lay the first egg. The first egg is always a male. And then as that pupa develops, you can see here, you notice here more and more Varroa offspring. So that by the time the adult bee emerges, that one Varroa mite that infested the cell can have up to 3 daughters. So it's not a high rate of reproduction, but just imagine this every time. They go out, they come back in, they come out, they come back in. They keep doing this until those numbers explode in your colonies. So why are Varroa bad? So remember I showed you that first slide where there was the Varroa mite right on the abdomen of the bee? Well, it turns out that's where they're feeding. So on the adult honeybees, this map here shows the frequency of where they observe Varroa mites feeding. And you can see most of the time they're feeding, it's on this part of the abdomen. The reason why they're feeding there is because there's an organ there called the fat body. And these are, all insects have them, but they kind of act like a Swiss Army knife in insects. This isn't just for honeybees, but all insects have fat body. If there's a physiological process, fat body does it. Protein synthesis, lipid synthesis, hormone synthesis, detoxification. It kind of acts like a liver in insects. And there's these certain proteins in that fat body called vitellogenin that they require to make eggs. So they must feed on the adults, specifically on this fat body, to acquire the nutrition that they need to lay eggs. It's kind of like how a mosquito needs to feed on blood. So that's what these Varroa mites do. They need to feed on fat body in order to lay eggs. So not only is that, you can imagine that feeding itself being pretty damaging, right? They're actively eating that organ from the outside and removing it, but they also transmit viruses. So when you're going into your bees and you see a lot of these bees with these deformed wings, these crumbled up, twisted wings, This is caused by a virus. And you only see these crumpled wings when the virus levels are very, very, very high. So we know that this deformed wing virus is directly related to the number of mites you have in your colony. If you see a lot of bees with this, you know you have a lot of Varroa mites. That's another way you could look. So they transmit that, and then they also transmit chronic bee paralysis virus. They'll typically look like this. They have these shiny appearance. Most of the time, bees are covered with hairs, but you can see these are kind of shiny, oily looking. And just like their name, the name of the virus implies, paralysis virus, they tend to move around a little slowish, lethargic, and things like that. So you could see these greasy-looking bees are affected with chronic bee paralysis virus. Again, all your bees are going to have these viruses, but in the presence of Varroa mites, the levels of those viruses go up very, very high. So if you see any of those two looking bees like that, deformed wings and these greasy ones, you probably have a high Varroa infestation. And so we know from surveys that Varroa mites are the #1 cause of colony losses. So this is an annual survey that goes out to beekeepers across the country, and they ask them, what are the reasons why your colonies died? So we have Varroa, queen issues, starvation, no losses, weather unknown, and a whole bunch of different reasons here. And they asked this to a bunch of beekeepers who are classified as backyard beekeepers. Typically, I think it's 5 or less honeybee colonies. Then sideliners are 5 to 500, and then commercial are over 500. So that's how it's broken down here. And you could see that most sideliners and commercial beekeepers say Varroa mites are why my bees died. So this is pretty important stuff. It's significantly lower in backyard beekeeping because I think that they just don't know about it, honestly. It's interesting to see when I go to these meetings how many people don't know what Varroa are and they don't know how to treat for them. So that's why I really want to reinforce today to start looking for Varroa mites, because once you don't know what you're looking for, you'll probably agree with the sideliners and commercial beekeepers. So most of the reasons why people think that bees are dying is because of Varroa mites. Well, that's just a survey, but what does the science say? And so these are a few papers that state this is what's happening. Look at this one. Varroa destructor is the main culprit for the death of reduced populations of overwintered honeybees in Ontario, Canada. Could it be any more obvious that that's what's going on? And so this is a really good one. And then this is a review paper showing how destructive Varroa are. So they looked at all this research that's been published in the meantime and showed that yes, all these papers are agreeing Varroa, Varroa, Varroa. And then we've even shown at our lab when we have bees that can resist Varroa, these pall line bees, we could show that they have higher survivorship. So this is what I really want to demonstrate, is that Varroa are a huge problem, whether it's here in the United States or across the world. And so when we think about Varroa, we have to think about a Varroa management system, because just treating with miticides isn't enough. Just doing cultural or IPM practices isn't enough. Using bees with good genetics, that's a good start, but it's not enough. We need to think about management systems as opposed to just any one of these things here, right? So we need to work them all together, just in case your miticides might fail. Maybe you got a bad batch or something, or maybe they're resistant to it. That's most of the research I do. So we have to kind of find ways to build redundancy in the system. And so the first thing we need to do is we need to know what Varroa look like. If you see this situation, this is really, really bad. You see the Varroa mites on the adult bees here? A lot of them. If you see that with your naked eyes on your bees, that's a high Varroa infestation. And typically, you'll see a lot more Varroa on the adults when there's no brood present. You see the lack of sealed brood here, open brood. So most of those Varroa mites are typically in the brood, but when there's no brood, they move on to the adults. And you can get a situation like this, that the adults are just covered with mites. That's a really bad situation. You'll also notice, too, some of the cells might have, right before they're sealed, can have a Varroa mite foundress invading those cells. Another good time to see these Varroa in the cells is when you're splitting your hives and you might have some burr comb between with some drones. That's another good spot to look for Varroa in the pupal cells. So you'll see them sometimes like this. Yeah, there we go. So that's splitting up that, so that's drones, and you can see here, just loaded up here with Varroa. All those little dots are Varroa mites. That's a pretty high infestation. And we'll talk about Varroa numbers a little bit later. So when you're looking at your frames and you're splitting your boxes, if you have any of that, that could be another good resource to look at. So those are just a little bit of just yes or no. They're not really quantitative, but now we're going to talk about a little bit more sort of quantitative measurements, so numbers. So one of the ways that you can measure the number of mites, and it's really important to measure because that'll tell you a lot of different things, is one of the ways you can do it passively is by using sticky boards. So on the bottom of the colony, if you have a screen bottom board, they typically have an area where you could slide a tray underneath it. And when those Varroa mites fall off the adults, they land on that tray that's covered with Vaseline, and they get stuck on there. So that's a good way to find out how many mites you have is by looking at that sticky board that's underneath the bottom of your colony. You could also put that, if you don't have a screen bottom board, you could kind of slide those in your normal hive bodies if the entrance is wide enough to kind of do this. There's a lot of different ways to do this. So this is a thing that you could talk about with other beekeepers is how do other people look for mites. Another way you could do that a little bit faster is by dusting the bees with powdered sugar. This does two things. One, it stimulates the bees to start grooming each other to knock the mites off. And also, too, the powdered sugar alone is enough to start knocking those mites off. So you could speed up the rate at which those mites are going to fall off the bees onto that sticky board by hitting it with a little bit of powdered sugar. What's the problem with using powdered sugar down here in Louisiana? Yeah, this stuff turns to like fondant in like 2 seconds if it's too humid out. So we could use powdered sugar. Like I said, we use this. We've also used potato starch, corn starch as well. They're all about the same. I actually think powdered sugar, it's too hygroscopic and it can absorb a lot of water, a lot of moisture. So we also have been experimenting around with potato starch and corn starch too. So anything with that kind of powdery area should be able to do it. Knock those mites off for you. Okay. All right, but the way that we mostly measure mites for scientific reasons is by doing these jar tests. And so the first thing that we do is you need these very expensive, high-tech pieces of scientific equipment, buckets, $5. And so the way that we do this, there's 1,000 different ways to do this, but I'm just telling you the way that we do it, so ask around. So what we do is we get a frame or 2 frames with sealed brood on it. That way we make sure we're sampling the same part of the colony every time. And then we just get it, hold it above the bucket, and just snap it like that. The bees will fall off into that bucket. After a few minutes, any older foragers will fly away, and then you could sample the bees in the bucket. It's really important that you don't sample the queen. This does happen. We try not to, but it still happens. So before you go to shake off that comb, make sure you don't have the queen on there. It's a good idea to find the queen, cage her, or put her somewhere else so you make sure you know she's not on there. That's always drives me nuts when I get these jars and I'm shaking. I'm like, oh no, there's a queen. So be careful not to sample the queen. All right, now we got our bees in our bucket. How are the ways that we can get them off? A lot of different ways, you can imagine that. So powdered sugar shake would be, I'm gonna use this one. So this is just a mason jar and a lid. This lid has a 1/4-inch screen on it. This is like my duct tape. I use this for a lot of different things around the lab, okay? So just the screen, lid, like this. You put the bees in the jar, give them like a tablespoon, maybe 2, powdered sugar. And you don't need to be really rough on them. Just kind of roll them around, get them all nice and covered with powdered sugar. Usually about a minute will do it. And then what we like to do is put it over a tray. And then you can just shake the mites and the powdered sugar out. The bees will stay in the container, and they'll go onto a tray here. If you're really good, you can kind of see a few Varroa mites here. But a way to really make it easier to see them is just get like a spray bottle, spray down the powdered sugar. That melts away, and then the Varroa are just left behind. So powdered sugar wake. This is a good way to use to sample them because you could just put those bees back in the colony, and they'll be fine. So you don't kill any bees with the powdered sugar shake. But you could knock them off of them with the powdered sugar, yeah.
Do they eventually consume the sugar, or is it?
Yeah, they'll group it off of each other, yeah. But the thing is, there is some research that came out 2 years, what year was that? 2 years ago, where if you shake them really hard with the powdered sugar in there, it will result in a lot of the bees dying. But we just like I said, just turn the jar over, gently roll it around. The powdered sugar is enough to start knocking them off. You don't need to really shake them really hard. Good. But the way that we mostly do it at the lab is by using detergent or alcohol. So we use a couple different things. So when I'm out in the field and I'm doing some of my testing, I'll put my bees in this container, and then you add water about a third of the way, and just 1 or 2 drops of Dawn dish detergent. It could be anything really. We just use Dawn because it's just what we do. Not that much, because you don't want too much foam. Too much foam the Varroa mites will get stuck in the foam and they won't come out. So there's a little bit of soapy water, about 300 mites, 300 bees. You put a solid lid on, shake that. See, these bees are dead, so you're not going to save them. These are what you're sacrificing to get your mite wash. Shake it for about a minute. Take that solid lid off. And guess what I do? Screen, see this? Again, we have thousands of these. This is a wintertime project. You could then put that screen lid on, pour the mites out of that container. The bees stay in there. Mites go in here, and you can count your number of mites. Okay. We use soapy water just because it's easier access and we don't have to. It's not hazardous. You could use alcohol. People use what's it called? Windshield wiper fluid sometimes too. You could, but it's just so much easier to use soap. There's no difference between them, so I just use soap because it's easy. You don't have to do this, but what we do at the lab is that when we take our sample, after we wash the mites off of it, you see the bees here in this tray, mites in the bottom. We count every single bee in that sample. It's a lot of work. Typically, if you use a half cup sample like this and get about 3 ounces of bees, that's about 300 bees, plus or minus. We have data from over the last 5 years that show, fill up that 3 ounce line, it's about 300 bees. 100 bees per ounce, rule of thumb. So it's really important to get only about 300 bees for 2 reasons. One is because that way you have enough bees to make sure you have a good representative sample. We don't want to sample too many because if you sample about 500-600 bees, that could be half a frame of bees. So you don't want to slow down your colony by oversampling. So 3 ounces of bees is about 300 bees. Okay.
Do you have any experience with the CO2 method?
Yes, we just got this in, and actually I don't think I have this on the next slide because it's an old slide. Yeah, so CO2 container. So this is sold from Man Lake by Vito Pharma. This is called the Varroa Easy Check. It's this container here, and it's kind of versatile. You could put your bees in here, soapy water, shake, and then you'll see the mites come down here on the bottom. So there's this internal basket, right? So that's one way to do it. You could also do powdered sugar shakes in here too, kind of versatile. But the way that we're going to test this out is that they also sell this CO2 syringe applicator. So you put your bees in here, and you crack the side. 1, 2, 3, 4, 5 seconds of CO2. CO2 will knock mites off. We've done that in the lab. 5-second shot, about 15 minutes, kind of just shake the bees a little bit, and the mites should fall through the bottom there. So we haven't validated that. We're going to do some data on this to make sure this works. But we know that in the lab, I was using CO2 to knock mites out so I could transfer them onto bees. Turns out that they die exposed to the CO2. So this method should provide another layer of versatility. So what I want to show from all of this is that there's a lot of different ways to look for Varroa mites. Now you have no excuse not to be checking for Varroa mites. Now you know how to do it. There's no wrong way to do it. You just have to find out how many mites you have. And for what you're doing, you're just going to look at the number of mites. You don't need to count all the bees and everything like that. Just find a way that you can quantify the number of mites. We've got a couple reasons. One is because it's going to tell you when to treat. So different times of the year, there's different mites in your colony. And I'm just going to standardize it by the August threshold, right? So typically we say August because with the commercial beekeepers we work with, that's when they start pulling honey. As soon as that honey comes off, treatments go on. And so when we look at these different ways to measure number of mites, we kind of have a treatment threshold. In fact, I think that this is an old presentation. We're going to have to revise this because we're finding out that sometimes this needs to be even lower depending on what stock of bees you use. So if we're looking at the sticky boards, we're saying that in August, if just 5 to 10 of them fall off onto the sticky boards, that's probably enough to warrant a treatment. If you're doing a sugar dust drop, you could do that in 1 hour. Remember I said it stimulates them to groom and knock off the mites. You could do a sugar dust drop and check that sticky board, and an hour later, 5 to 10 mites, that's probably enough to treat. The sugar shakes take about 5 minutes from start to finish, and if you have 3 of them in your sample, that's probably enough. And then finally, too, with the detergent shakes, about 3 mites in that sample. So you can see here it's not a lot. And the reason why it might not seem like a lot is because there's there's many more mites in the brood. Typically, rule of thumb is twice as many mites in the brood. So you can think about, if this is what we're seeing on the adults, when there's brood present, most of those mites are in the brood. So you can see here different thresholds for treatment. And the reason why is 2 things. One, mite populations aren't always synced up with the bee populations. And this will make a little bit more sense. So let's say this is the beekeeping year from January through December. And on this axis, we have the number of bees in the colony. You can see here by this green line. And then this axis here is the mite population by this light green line. So as you can see here, when we're in April, colonies are doing good. The adult bee populations are going up and up and up and up. And then typically here around June is when they'll start to peak. That's after the tallow flow. So it's getting too hot and dry. And so that's typically when honeybee populations peak. And then they decline gradually throughout the year until they start laying less brood and the colonies just naturally decline and dwindle. But you can see here, Varroa mites, their populations are growing a little bit less quickly. And their populations actually peak in August as opposed to June. So you might have a lot of bees in your colony, but very few mites, and think, oh, this is great, everything's fine. But you can see this large peak coming up. So as that peak comes up and the adult population starts to decline, you'll start to see more and more mites on your adult bees. So this is why you have to kind of think about this. There's not one number to rule them all. You have to be really paying attention to your mite levels throughout the year. The lower you could get them throughout the whole entire year before they get high, I could guarantee you'll lose fewer colonies. So that's why these thresholds are going to change every month. And that's why I indexed it to the August mite peak, because that's when mite populations are going to probably peak here in our bees.